So let's start the session. This is week 6 online live session for the NPTEL course on Urban Utilities Planning, Water Supply, Sanitation and Drainage. The course instructor is Dr. Devapratim Pandit sir. He is a professor in Department of Architecture and Regional Planning at IIT Kharagpur. I am the teaching assistant for this course. I am a research scholar in the environmental engineering part in the Department of Civil Engineering at IIT Madras. So this is the week 6 lecture outline. So in this lecture we will study the treatment of water and wastewater and what are the units and processes which are used for the treatment that we will see, and we will solve some MCQs, fill in the blanks and match up the following. In this lecture we don't have any numericals. So we will proceed with the questions. First we will proceed with the flow diagram, like how the treatment of water is carried out. It is a general treatment flow sheet, so that we generally follow. Treatment of water. So first of all, the water is taken from the rivers with the help of intakes. So after intake, so it may have intake may contain screens also. So screens are provided in the intakes to prevent floating materials, and then after screens, the water is the water may undergo aeration. Aeration will remove the dissolved gases, and if it is from groundwater, iron and manganese can be precipitated with the help of oxidation. So in aeration, the water is brought in contact with air, and air helps in oxidizing the reduced substances, and it then it will precipitate iron and manganese, and few dissolved gases which are already there, that also can be removed with the help of aeration. After aeration, it will contain some dissolved solids. After aeration it will contain dissolved and as well as suspended solids. So first we will remove the suspended solids. So suspended solids can either be removed with the help of plain sedimentation, either with the help of plain sedimentation, or we can go for coagulation, flocculation and sedimentation. So, or we can go for coagulant aided sedimentation. Then after this, treatment, we can go for, so till here we have removed suspended solids. The screens, we have removed big floating solids. Aeration, we remove in aeration we remove dissolved gases, and iron and manganese. And here we remove suspended solids. And after this, we can go for filtration, because filtration can remove small colloidal solids. Filtration will remove small colloidal particles, and after filtration, we can go for disinfection. After filtration, we can go for disinfection. And disinfection will kill the microorganism, and then we can further use the water. And there is one more thing. After disinfection, Disinfection. If water contains hardness, we can go for softening of water. Softening of water. That we can do with the help of lime soda method. If it has salts, then we can go for desalination. If it has more fluoride, then we can go for defluoridation. So likewise, if water has some excess, if water has some excess uh, pollutants, then we can remove with the help of these units, which are in this slide and which are in the previous slides. Are you able to see my slides? You can text. Are you able to see my slides? I can't hear. Okay, I will proceed. So this is our treatment flow sheet. I will repeat it. So from intakes, we will take the water. So from intakes, we will take the water, and in the intakes itself, we provide screens to remove the floating solids. And after screens, we may provide aeration sometimes. Mostly aeration is provided for the groundwater, not for the surface sources. So with the help of aeration, we can remove the dissolved gases and iron and manganese. After aeration, we can either go for plain sedimentation and then and or we can go for coagulant aided sedimentation. In this coagulant aided sedimentation, we add the coagulants, which helps the particles to agglomerate. So particles will come in contact with each other and they will make a flock and then they will settle in the sedimentation tank. While in plain sedimentation, a tank is provided without any chemicals, so the particles will settle in a discrete manner, and it will not the particles will not affect each other and they will settle at the bottom, and from the bottom the sludge is removed. And after the sedimentation, the water will go for filtration. So filtration will remove the small suspended particles which are not removed in the sedimentation process. After filtration, we provide disinfection to kill the microorganism. So once the microorganisms are killed, we can use the water, especially for drinking. So disinfection is mainly provided for the drinking purpose, to kill the disease-causing bacteria, because most of the diseases are due to the water. And after disinfection, we may provide some provision. Suppose if our water is hard, we may go for softening of water. And if it contains more salts, we can go for desalination. If it contains more fluoride, we can go for defluoridation. So it depends on the water or source of water, that what extra constituents or which pollutant does it contain. So based on that, we can modify our this treatment flow scheme. So now let's start with the questions. So palatable water is tasteful for drinking and The answer is A, aesthetically pure. And one more thing about about this actually potable water, it means it is safe for drinking purpose. Means it meets all the standard. Colloidal impurities in water are of size. What is the size of the colloidal impurities? So there are, if I divide the part solids are in the range, we say solids are suspended solids, and then we say dissolved solids, and there is a range in between, that is these are called colloidal solids, and these solids will impart turbidity. Okay, and the size range is in between suspended and dissolved. So their size range will be in 10 to the minus 3 to 10 to the minus 6 mm, like this, minus 3 to 10 to the power minus 6 mm size. This is their size. Here is one more question. Match the following. So here we have the impurities, and what are the processes used to remove those impurities. So colloidal impurities can be removed with the help of colloidal impurities can be removed with the help of coagulation. P3. Suspended impurities, sedimentation. Q1. Dissolved impurities like salt in water can be removed with the help of evaporation. R3. P3, Q1, R. Wait. Evaporation is 2. Evaporation is 2. So 3, 1. So answer is C. So answer is C here. Inorganic salts, organics and gases in soluble form are called inorganic salts. Inorganic salts like NaCl. Organics like, let's say, glucose, sugar. Gases like CO2, ammonia. These are in soluble form. These are called, what we'll call, if these are dissolved, these are called gases. We can not include here, but these are called dissolved solids. If we exclude these things, so let me change this. So this will be called as dissolved solids. Excluding these are called dissolved solids. What is the meaning of representative sample? So if we are checking the quality of water or wastewater, let's take an example of lake. So what is the meaning of representative sample? So here I will give you an example. So this is the sewer, let's say. Sewer is polluting a lake. This is the lake. So sewer is polluting, let's say this is the influence. This is the influence due to sewage pollution. So if I take sample from location A, if I take location from sample A or B, will it signify the pollution? No, it won't signify the pollution. It won't signify the pollution. So uh, if you take the example C here, it may signify you the pollution. So C can be taken. C at location C, At location C, C can be taken as representative sample. Second, give example. C can be taken as representative sample. So while we, if we take, if you take sample from A and B, the influence of sewage pollution is not counted. So that's why it is suggested to take sample nearby the sewage pollution. So this is called representative sample. Sample A and B are not considered as representative sample. Samples should reach a laboratory within how many hours of collection? Sample should reach within 72 hours of the collection. So in laboratory, we have to do the analysis within 72 hours. For sampling of water wastewater, minimum volume sample requirement is, the minimum volume is 250 ml. Rates of chemical, biochemical and biological activity are determined by rates of chemical. This will depend on temperature. But not only it depends on temperature, it may depend on any other factor. Temperature, pH, dissolved gases like carbon dioxide, hydrogen sulfide, chlorine, and oxygen carried out. They are carried out instantly on the spot. The guidelines, the guideline value of E. coli 0 per 100 ml is for. So E. coli is an indicator, is a yeah for the pathogens. So that should be actually 0 per 100 ml. That is that this is for all water intended for drinking, treated water and treated water entering the distribution system, and treated water in the distribution system. So the water which is intended for drinking and the water which is sent to the distribution system, so after the treatment it should be 0. Color due to dissolved solids is called. So we have color. So the water can have color due to, let's say the water can have color due to dissolved solids and suspended solids. Okay, so collectively they both will impart color. So the color which is imparted by dissolved solids, this is called true color. This is called true color. So and the color which is imparted by both is called apparent color. The color which is imparted by both is called apparent color. So color due to dissolved solids is called true color. Correct? The specific conductivity of water can be used to estimate the total amount of, so specific conductivity. So the conductivity, first we need to understand what is conductive, how the water is conducting electricity. So this conductivity is electrical conductivity. The conductivity in water is due to the ions. Ions will move and then they will cause electrons to flow. So these ions are due to inorganic solids. Inorganic solids. What are inorganic solids? Let's take an example. NaCl, KCl, these ions, they will conduct electricity. They will conduct electricity. But so uh, inorganic solids will conduct electricity, but organic solids do not conduct electricity. And this we are talking about dissolved inorganic solids. So dissolved inorganic solids like NaCl, KCl, and other salts, they will conduct electricity. So here, if I want to say, so I will take one more page. Insert. So dissolved solids, if I talk about dissolved solids, so dissolved solids can be organic, can be inorganic. So in water supply, organic solids this concentration is low. Water supply, if I say total dissolved solids, that will be organic plus inorganic. So organic content is low, organic content is less. So that's why TDS here is equals to inorganic solids, inorganic dissolved solids in water supply engineering. But in wastewater engineering, we have human excreta and other biological waste like from sugar industry, paper pulp industry. So there we can't say TDS is equals to inorganic solids. So here we can say if specific conductivity of water can be used to estimate the total amount of TDS, total dissolved solids, because organic fraction is less. Okay, so this is the assumption. Uh, I hope you are getting whatever is uh, means whatever I am discussing here. You can text if you have any doubt. Name some waterborne diseases. If water is not properly disinfected, or if the so in the distribution system, so let's take this example again. So from this distribution reservoir, this is a distribution reservoir. Let's take this is an overhead tank. This is the overhead tank, and it is distributing the water to a city. This is the distribution system. These are roads and all, so I'm not making it properly. So if there is any leakage in the joints, leakage in the pipes and joints. So, and these pipes are laid below the ground, and sewers are also laid below the ground. And if there is leakage from sewage and there is mixing of from sewers, so there is mixing due to sewage. If there is mixing due to sewage, so then the sewage may mix with the water supply, and it may cause diseases sometimes. So that's why most of the diseases we face are mostly due to the water also. So the diseases which are from water, which and these are cholera, typhoid, fever, dysentery, hepatitis, cardiasis. These are the waterborne diseases from which if water is not disinfected properly, or even after the disinfection. Let's take an example. So this is after disinfection. So after disinfection, the water is supplied to the overhead tank, and after overhead tank, it is sent to the distribution, sent for the distribution here. So if any mixing happens with sewage or soil also, so in soil has the bacteria. We know soil consists of bacteria, viruses, many things. So if intermixing happens, so then it may cause diseases to humans. So that's why we need to take care of this. How we will take care? So what we will do? So that's why we say access chlorine. Access chlorine means we provide, we should maintain, we should maintain residual chlorine, residual chlorine. We should maintain residual chlorine after the disinfection. So this residual chlorine so means you may feel like after, even though we get the water after the treatment, it has excess chlorine. It will taste bitter because of this excess chlorine. That is called residual chlorine. So this will kill the microorganism which can affect us, which can affect like disinfection is already done here. So in this unit we will provide some extra chlorine so that it will kill the microorganism which may enter in this distribution system in future. So that's why some extra chlorine is also provided. The range is 0.0 to 0.3 milligram per liter. Why biological tests are performed in water? So as we discussed just now, like all these diseases are from water. This here we discussed. So because of the diseases, because uh, of these diseases which may affect our living, day-to-day living, so we need to perform the biological tests. That's why we perform. So living organisms like bacteria, virus, protozoa are infectious to humans and they can affect the human health. So that's why we need to perform the biological tests. What is BOD5? BOD5 is biochemical oxygen demand of water during first 5 days at 20 degrees Celsius. This is the standard. So BOD will tell the strength of wastewater. Like if BOD is more, like in 5 days, if oxygen consumption, must understand it basic. We'll take the we'll take the wastewater. We will add diluted water or shaded water. And then we'll put some buffers, which is yeah, let's not discuss it now. So these are the BOD bottles. This is the wastewater for which we need to calculate the BOD. This is the diluted aerated water. Aerated water. Aerated water. So it has no dissolved oxygen, no dissolved oxygen. It will contain saturated dissolved oxygen. It will contain saturated dissolved oxygen. So the microorganisms are also provided, and it will be kept for 5 days at 20 degrees Celsius. So this oxygen here it will be oxygen, here it in the wastewater will not contain oxygen. So the microorganism will consume oxygen and they will degrade the organic matter. And so the oxygen concentration we will calculate it first. So DO initial will calculate, and after 5 days also we will check the DO value. So this will be called as DO final value. So the DO which was initially there in the bottle before 5 days will be reduced. So more is the difference, more is the organic matter. So the microbes will consume the oxygen and the microbes will decompose the organic matter. So more organic matter is, if more organic matter is decomposed, then more oxygen will be consumed. So by this way we can calculate the biochemical oxygen demand, and if more the difference, more the BOD, and more is the strength of wastewater. It tells the organic strength of wastewater, or we can say water. Winkler's titration is used for. So the Winkler's titration is used for dissolved oxygen. So here the BOD test, initially if we mix the aerated water with the wastewater, we will check the oxygen, this DO, dissolved oxygen, we will check with the help of Winkler's test first, and at T is equals to 0 and at T is equals to 5 days, we will check the DO final using the help of Winkler's test. Now we use modified Winkler's test. Yeah. So, but it takes 5 days. So the disadvantage of this BOD test is it takes 5 days. But if we need early results, we can go for COD method. Presence of nitrogen in water indicates. So present in nitrogen, it indicates organic matter in water and also indicates the presence of bacteria. Okay, what is the doubt? Sir, kindly explain once more. If more BOD, then more impurity. Yeah, correct. If more BOD, it means more organics, more organic matter. Okay, I will explain it once more. So you understand what in the BOD test microbes are used. So microbes will consume oxygen and you have organic matter. And what will happen? Microbes in presence of oxygen will degrade the organic matter and will oxidize the organic matter in presence of oxygen. So you will get CO2 and the microbes will grow and then we will get H2. So what is happening? So if more is the organic matter, more oxygen will be required to oxidize it. Just consider it as simply as more organic matter will require more oxygen requirement. So that's why if more organic matter, so more oxygen is required. Simply we can say it has more biochemical oxygen demand. That's it. I hope you understood it now. Is it fine now? So the nitrogen in water is available in four forms. So first one is organic nitrogen. Second one is ammonium nitrogen. Third is nitrite nitrogen, NO2. Fourth is nitrate nitrogen. So organic nitrogen and ammonium nitrogen both are called Jaldal nitrogen. It is called TKN, total Jaldal nitrogen. So yeah, now let's see the question. So we understood like nitrogen, what will nitrogen indicate? So nitrogen will indicate the presence of organic matter and presence of bacteria. So this is the oxidation state, means that you will understand it later in this question. So free ammonia. So we have to solve this question. So free ammonia, what it indicates? Nitrite, what it indicates? Nitrate, what it indicates? So this is called nitrate. This is called nitrite, and this is called free ammonia. Okay, and this is called organic nitrogen. So free ammonia, nitrates, organic carbon. I think it's organic nitrogen. Yeah, it's organic nitrogen. So nitrates. So if you get the nitrates, let's take one more example. Suppose this is the river. If this is the city, there is a city here. City is discharging the sewage. After some time at the so naturally the sewage will be treated. After the treatment, you will get nitrate. You will get nitrate. If you get ammonia here in between NH3, it means it is partly decomposed. Partly decomposed. If you get nitrite here, NO2, it means decomposition has started. If you see organic nitrogen, see these are along the length. Consider it like this, along the length. So along the length, you may find organic nitrogen and ammonia at the same place. These two things you can find nearby. But if the water will flow, so it will be oxidized to nitrite, and then further if it will flow means it will with respect to time, with respect to along the length and time. So with time, the decomposition of organic matter is going on, and the oxidation of organic matter takes place, and along the oxidation of nitrogenous matter is also taking place. So the organic matter, this ammonia, is then oxidized to nitrite and then nitrate. So nitrate will tell us the organic matter has been decomposed, has been organic matter is fully degraded. Ammonia will tell it's let's go to this question. So nitrate will tell us fully decomposed, or oxidized organic matter. So these three will be first stage of decomposition, recent contamination. So yeah, here I need to make one little change. Nitrate will this will indicate fresh contamination. Indicate fresh contamination. And ammonia, this nitrate will tell partial decomposition. Partial decomposition. Now I think it is clear. Nitrite will indicate, free ammonia will indicate first stage of decomposition, recent contamination, that is D. Nitrites will indicate partly decomposed, oxidized organic matter, that is C. And organic nitrogen will tell the quantity of nitrogen before decomposition, that is A. So if we can see the sequence, here we should, let me write this. So this organic nitrogen will then be converted to ammonia, and then it will be converted to nitrite, then nitrate. Organic nitrogen, this is free ammonia, nitrite, nitrate. Fully decomposed, partly decomposed, recent pollution, decomposition has not started, decomposition not started. Okay, so these forms of nitrogen signify these things. Any doubt here? Okay, now we'll move on to the next question. Starch iodide test is used to detect chlorine. So what is happening here? The chlorine in water. The chlorine in water. We will put starch in water and then we will put iodine in water. Not iodine. We will put starch. We will put potassium iodide. The chlorine the chlorine will react with potassium iodide and it will form iodine. This is called iodine. And this iodine plus starch. So if we have more chlorine, we will get more iodine I2, and if I more more iodine will react with starch, it will give us blue color. So more blue color is observed, and to quantify this, we will titrate it with sodium thiosulfate. And if more volume of thiosulfate, if more blue color, more thiosulfate will be consumed, and more volume will indicate us more chloride content. So that's it. Now, next question is, what is hardness? So, the hardness is due to the multivalent cations. What are those multivalent cations? Calcium, magnesium, aluminum, and if these cations, if these are with the anions like CO3 2 minus carbonate and bicarbonate, this is called carbonate hardness. And if these cations are with nitrate, sulfate, and chloride, this is called non-carbonate hardness. Carbonate hardness means if you boil the water, the if you boil the water, you can see the precipitate. This hardness, this this hardness you can find at the bottom of the flask. And while this non-carbonate hardness that can be removed with the help of heating. So this is called non-carbonate hardness, and it is also called as permanent hardness. Permanent hardness. This is called temporary hardness. So we can also define it as hardness, the water which can't make leather, which can't make sufficient leather or foam when mixed with soap due to the presence of calcium and magnesium salts, which forms come by the reaction with soap. So due to the hardness, we can't make the sufficient foam or leather. That is called hardness, and hardness causes greater soap consumption, scaling of boilers, causing corrosion, etc. So temporary hardness is due to carbonate and bicarbonate that we discussed. The solution is boiling or addition of uh, by adding the lime to water. This is first thing. Sulfate, nitrate, chloride of calcium and magnesium require water softening, that is known as permanent hardness that we discussed. Hardness is usually expressed in milligram per liter of calcium carbonate in water and determined by Wasserate method or EDTA method, EDTA solutions. That we will not discuss it now. Hardness up to 75 ppm are considered as soft. If more than 200 ppm, it is called as hard water. Temporary hardness in water is due to nitrate will impart permanent hardness. Chloride will also impart permanent hardness. Carbonate will impart temporary hardness. The answer is C. Chlorine is usually found in water to disinfection of water using chlorine. What is the unit of turbidity in water? TCU is true color unit. True color unit. This is for color as the name suggests. TON is threshold order number. This is for order, as the name suggests. NTU is nephelometric. NTU is nephelometric turbidity unit. So this is for the turbidity. ppm can be used for any other thing. So NTU, this is for this measure using. So the turbidity is, as we discussed, about colloidal particles. Colloidal particles will cause turbidity, and suppose this is the sample. This is the light source, and at 90 degree we have the receiver. At 90 degree the scattered light so whatever the light received at 90-degree angle, that is collected here, and This gives us the reading of NT. And for a standardization, we use a chemical. Its name is formazine. So what you will do here, you will take the water sample inside, and you will and in the instrument itself, the light source will send the light, and at 90-degree, the whatever the so here we have diodes, photodiodes. The diodes will take the light and will consider the light intensity, and they will give the results in the form of signals. So that will be converted into NTU later. Which of the following instrument helps in detecting the color of the water? Turbidimeter is for the turbidity, thermometer is for temperature, conductivity meter is for dissolved solids which are inorganic in nature. Photometer will give us the color value. Treatment of water depends on. So if you are going to treat the water, how much you need to treat? How will we know? So it depends on the quality of raw water or available water, and what is the quality of desired water. Quality of desired. Quality of desired water. So if there is any significant difference in between these two, then we will go for treatment. So the quality of available water or raw water and the intended use. So if there is any significant difference in between these two, then we will go for the treatment. Match the following. So here we have pollutants and we have the processes. Processes. So dissolved gases can be removed with the help of aeration. Microorganism and colloidal matter. Okay, let's see this first. Suspended impurities are silt, clay, sand that can be removed with the help of plain sedimentation. Is third. And microorganism and colloidal matter and fine suspended matter. So now two left. One, three left overall. Two and four. So filtration, fine suspended matter can be removed with the help of filtration. Fine suspended matter can be removed with the help of filtration, and microorganism and colloidal matter can be removed with the help of coagulation, flocculation, and sedimentation. So one more thing. So colloidal particles These are very small and we call them stable. Stable. Stable because and we make them destabilize with the help of coagulants. The help of coagulation we destabilize them. Stabilize. So stability here means stable means they have charge and they repel each other and they will undergo Brownian motion. Because of this, these all things, they don't agglomerate. They cannot agglomerate. Agglomerate means they cannot combine. And if they can't combine, they can't make a big flock and they can't settle. So destabilize means their charge is neutralized. Charge is neutralized and sticky, gelatinous compound is used to agglomerate which takes a big particle for better settling. That's it. For better settling. Which of the following treatment method is primarily used to remove pathogenic bacteria in the water treatment plant? This is disinfection. In water treatment plant, to which unit does not does water move from flocculation basin? Answer is settling basin. In water treatment plant, to which unit does water move from flocculation unit? So after flocculation, like I already discussed, we add the coagulants. We add the coagulant. We provide rapid mixing. Then after rapid mixing, the water goes to a flocculation tank. There we provide slow mixing, and then after it goes to settling basin. Coagulation, rapid mixing, flocculation, slow mixing, settling basin, settling occurs. Which of the following is correct sequence of treatment units for the highly polluted surface water? So if you see A and D, only pre-chlorination is extra here. And this is the sub part: sedimentation, slow filtration, disinfection, rapid mixing, flocculation. But sedimentation is missing here, so that's why it is wrong. Sedimentation, slow filtration, disinfection. Third, this is not complete because we are providing it for the highly polluted water. This is very less units are provided. So in rapid mixing, flocculation, but sedimentation tank should be provided. Otherwise, there is this in the filtration. Filtration will be clogged. That's why B and C are incorrect. And in now the competition is between A and B. So the water is highly polluted. So if the water is polluted, it may contain organics also. And if organics are present, so the organics may form, they may cause taste and odor. Taste and odor. To remove taste and odor, we'll pre-chlorinate the wastewater. Pre-chlorinate wastewater. And if it contains high pollution, the microorganism may also further grow. So in order to reduce the microbial growth, reduce the microbial growth, we'll, we'll provide chlorine so that the pipes and the units which we are already which are already there, they should not affect the treatment. Like pipes may have microbial growth and other units may have microbial growth, then it can affect the treatment efficiency. So that's why we provide chlorine before disinfection. Also we in at disinfection also we can provide the chlorine, but in order to increase the efficiency for all the units, we can pre-chlorinate also. After that, this aeration, flocculation, sedimentation, filtration we have discussed in the first slide itself. So the answer is A here. Which of the following factors do not influence plain sedimentation? Plain, there are four types of settling. Four types of settling. So the plain sedimentation is type 1 settling. Type 1 settling. This is called discrete settling. So in discrete settling, there is no change in shape, size, or weight of the particle. So the answer is C here. Change in shape, size, or weight of particles during settling. The rate of exchange of gas in the process of water purification depends on. So the rate of exchange of gas in the process of water purification depends on area of interface between gas and liquid. Yes, correct. Suppose this is the tank, aeration tank, and this is the surface area. Will affect area of interface between gas and liquid. Yeah, it is. So the amount of gas entering will depend on the surface area. So it is correct. Partial pressure of the gas, yes, it depends on the compression depth and time of contact. Yeah, time of contact is also required. Compression depth is not. Let's check. Compression depth is P, Q, and S. So it depends on this, and this. The rate of exchange of gas depends on the area of interface that we discussed, thickness of the interlayers, time of contact, time of contact, uh, time partial pressure of the gas, and degree of undersaturation and oversaturation of the gas. So let's take an example. So if oxygen is less here, so oxygen will come from the atmosphere, and simultaneously, because the microbes will consume oxygen and they will produce carbon dioxide, it will go to the atmosphere. So the flow of gas depends on whether the oxygen is undersaturated or CO2 is oversaturated. So in this case, the CO2 is produced because of microbial activity, so it will go into the atmosphere, and oxygen is less, so oxygen will come from the atmosphere. Do you have any doubt? Match the type of aerators with the appropriate description. So spray aerator. For spray aerator, they will provide mist or droplets. Diffuse aerators, compressed air is used. Cascade aerator, water passes over steps. So in spray nozzles are like water is sprayed. Diffusion aerator, diffuse aerators. From bottom it is diffuse. Diffuse aerators. And in cascade aerators, the water will flow from the steps. The water will flow like this. So the thin layer forms and that will cause aeration. It's called cascade. Supply. In disinfection of water, chlorine demand is the difference between dash or remaining chlorine in the water after a specific period. So we want to know what is the chlorine demand. So how much we need to know how much chlorine is added. 20 milligram per liter, and remaining is 5 milligram per liter. Difference is 15 milligram per liter. That is called chlorine demand. Chlorine demand added minus remaining. This is added minus remaining. This is the chlorine demand. Which of the following are additional treatment performed in water treatment plant based on the unique characteristics with respect to the location? So in the first slide we studied some things. So segregation is for solid waste. Solid waste. Incineration is also for the solid waste. So rest fluoridation, we add the fluoride because fluoride is helpful in and the for the for our teeth. So fluoride reacts with the tooth's enamel and give more strength to the teeth. So that's why we need to add the fluoride. So fluoride is also added into the toothpaste. But if you provide more fluoride, it can cause fluorosis also, and if the fluoride concentration is more than 5 milligram per liter, it can cause bone fluorosis and the bones become brittle. So excess fluoride should not be there. Distillation and accurate carbon is used for adsorption. So P, R, and S. R and S is the answer. Gas promotes exchange of gases between water and atmosphere. Aeration. What are the uses of aeration process? So it removes dissolved gases which cause taste and odor. And dissolved gases like carbon dioxide, hydrogen sulfide can precipitate impurities, iron and manganese that we discussed in the first slide. Second slide. Factors influence sedimentation. What is sedimentation? We'll just study in general. Sedimentation is nothing but suspended solids which are in water. If we reduce the velocity of water, they will settle with the help of gravity. This is called sedimentation. So, so the sedimentation depends on the what is the density of water, what is the viscosity of water, what is the size of the particle, the dia of the particle, and what is the yeah. So dia will tell us what is the settling velocity of the particle. It depends on some factors. And what is the concentration of these? What is the type of solid also? Type of solid. So we'll study here. So it depends. Factors which influence sedimentation is size, shape, density, the density of water as well as solid. Because if the density is more, then only it can settle. Otherwise, it will not settle. Viscosity, density, and temperature of water. Surface overflow rate, that means settling velocity of the particle. Velocity of flow. If the velocity is very high, the water is entering here and water is going out, then particles will not get time to settle. Inlet and outlet arrangement, detention period, how much is the time for settling, effective depth of the settling zone, if this depth is sufficient or not. Lime soda process is used to remove, so it is used to remove the hardness. What is chlorine demand that we already discussed? Chlorine 20 is applied and 5 is remaining, so 15 is the chlorine demand. Added, this is added and this is remaining. So rest is chlorine demand. Can excess lime be used as a disinfectant? Yes. Yes, it can be used as a disinfectant. Yes, it can be used as a disinfectant because excess lime kills bacteria by raising the pH. Because the pH increases, pH increases till 9.5. So, and microbes can't survive beyond this pH, so they will die. So that's why it can be used as a disinfectant. Here we have one more question. This is the question. Aeration is process promotes exchange of gases. Process promotes exchange of gases. Filtration is chlorination is addition of chlorine to the disinfect water. Filtration is process of removing suspended matter using sand. Sedimentation is settling of heavy particles before filtration. This is the last question of today's session. Thank you. I think we can close today's session. Thank you all for the joining.